Okay, hello everyone. My name is Ismayl. I'm a year 1 student here, and I'll be presenting for you the lymphatic system, and then my colleagues Noor and Yasser are going to explain the immune system. Inshallah, you'll find it beneficial and it will be useful, inshallah. So it's mostly questions for you to practice, and it's nice to see you all like fresh after the vacation. Of course, resting is a priority, but I hope you studied 10 minutes at least in the holidays. So did you study lymphatics or not? No? Okay, it's okay. It will be like a revision too. Okay, so you'll have a bit of background. We'll start with the first question. Which of the following statements is true about lymph? Produced in the kidneys, contains red blood cells, it transports dietary fats, or it's acidic in nature? Which one? You say C. C? Yes, it's C. So lymph, of course, is not produced in the kidneys. What's lymph? Basically, how it forms is that when blood is passing through the capillaries quickly, some of the plasma leaks out. Most of it returns back to the veins, but some of it remains in the interstitial fluid. If it keeps accumulating, and it's not exiting or draining, what will happen? Edema. We'll have swelling. That's why we need lymphatic system. And whenever we have edema, you need to put lymphatic system as differential diagnosis for it, because it helps in preventing edema. Red blood cells, obviously not. Red blood cells only stay in the blood. Transports dietary fats. You know which specific lymphatic pillary transports fats? Lacteals in the small intestine. They're important to know them, and the fluid inside of them is called chyle for fats, and it's not acidic in nature. Okay? Not a function. Which one is not a function of the lymphatic system? Absorption of fats, we just mentioned it. Transportation of hormones, removal of cellular waste, or immune surveillance. Yes, B. Function of blood. Hormones is for blood. And the others, yes, lymphatic system plays a role in immune system, which is why it's largely connected to the immune chapter. I advise you to study them together. And dietary fats, as we mentioned, lacteals in the small intestine. What's important to know is how lymphatic capillaries differ from blood capillaries. So try to answer this question. You say C. Okay, we have B also. C. Okay, let's go with them. Endothelial cells have tight junctions. Now we want plasma, we want the fluid to enter. We don't want it to stop it from entering. So of course not tight junctions. Oh, sorry. Many valves prevent the flow of fluid. We have endothelial cells are anchored by filaments to the surrounding structures, and they're open-ended. I'll show you a picture now to show you the characteristics, and then I'll tell you the right answer. So here we see the lymphatic capillaries. The endothelial cells overlap each other. They form mini valves. So fluid, when there's more pressure outside, there's more fluid, the valves open, it enters the capillaries. When it enters the capillaries, there's no pump, there's no heart for the lymphatic capillaries. So it's already low pressure. And it's moving slowly. So when fluid accumulates inside, it will exit. So that's why we have mini valves. They'll close, so no fluid exits. And it's also anchored to the connective tissue by filaments. And the last thing you see is that it's close-ended, not like capillaries are open on both sides, because they come from the heart and they go back to the heart. So here, it's between B and C. But the right answer is C. Why B is wrong? Look at it.
It says
into the lymphatic vessels. That's wrong. Many valves prevent fluid from exiting. We want fluid to enter. We don't want it to exit. So complete the choice till the end, and maybe they can trick you in some questions. So always read all the choices and the questions. This sequence is also important to know. See? Yes, it's C. We start with capillaries, small, then they form collecting vessels. Then we have trunks, then we have ducts. We have two lymphatic ducts. What are they? Thoracic duct and right lymphatic duct. So, yeah. Thoracic duct drains from where? Okay, think. And we have right lymphatic duct. Right is for, it will be right side. Okay, B, yes, it's B. And digestive tract, but they said digestive tract only no. It's also طيب the right lymphatic duct. Said D, B, D. Yes, D. Only the right upper limb, right thorax, and the head. Only this part. The rest is all thoracic duct. And here, this is a picture that can clearly show it. This is all drained by the thoracic duct. The right lymphatic duct only takes a small part. And we need to know, like from the book, the important points that in some individuals, the thoracic duct starts from something called cisterna chyli. It's found down here, like between the thoracolumbar area. And you just you need to know the name. Like it might come maybe as a direct question, oh, what does thoracic duct originate from? Cisterna chyli. And the ducts, where do they drain in the end? Our aim is to return this fluid back to the blood. So we drain back into the veins, the junction between internal jugular vein and subclavian vein. This is very important. You can see here the subclavian veins, and here you see this is the internal jugular vein, but it's not showing here. So it's the junction between them. The ducts drain the lymph back to the blood, to the veins. Lymph transport. When we talk about lymph, you compare it to veins, very similar, because it's under low pressure. You don't have heart. How will the lymph move? The skeletal muscles, like veins, contraction of skeletal muscles. You also have the pressure changes in the thorax. You also took that in the veins, movement in veins. Pulsations of nearby arteries, because you also take it in year one, that most of the time you'll have artery, vein, lymphatic vessel together in a sheath, connective tissue. So when artery is pulsating, also the lymph moves. And we also have smooth muscle in the walls of the large lymphatic vessels. They contract, but this is not present in the small lymphatic vessels. Important terms to know is you have the diffuse lymphoid tissue and we have lymphoid follicles. Diffuse, they're just here and there, almost in every organ, just a few lymphoid cells like lymphocytes and reticular fibers. Lymphoid follicles, they're like circle. They form follicles and they have important thing called germinal centers. So germinal centers have B cells. So whenever you know there's a lymphoid follicle, you'll know there are B cells present because of the germinal centers. And when do these increase in size? When B cells are activated. Okay, so these are important, they will help you later. Which of the following is the site of the first maturation of B cells? B. D, yes, bone marrow. So bone marrow, B for B cells, maturation of B cells. T cells where? Thymus, T and T. So these are called primary lymphoid organs. Now we go to secondary lymphoid organs. Anatomy in this, maybe the trickiest part of this chapter is the anatomy. So we have, you need to know the anatomy of lymph nodes, spleen, thymus, and malt. These are small parts. So I summarized them for you here, and it's always beneficial to put them together to compare them so that you know the features. So lymph nodes, here we have a picture of the lymph nodes. Lymph node has many exceptions that makes it different from other lymphoid organs. What is it? It's the only organ that cleanses the lymph. So it has lymph entering it, it cleans it, and it exits. Other lymphoid organs like spleen and all, they have only lymph exiting them because they're also like, they need to be drained from the plasma leaving. But they don't have lymph entering and getting cleaned and all of that. But the other lymphoid organs, they also play a role in immune function. They have lymphocytes and all of that. As well as lymph nodes, but lymph nodes cleanses the lymph, like it fights pathogens in the lymph. So that's important. So they have both afferent and efferent. Efferent E for exit. You know these are the vessels exiting. It has capsule. It has special capsule inward projections called trabeculi. It's made of cortex, which is outer area, this one, and medulla. Now in the cortex, they're important. You have follicles. What did I say about follicles? Germinal centers. What do they have? B cells. So you need to remember that. And this is present in superficial cortex. So superficial cortex has B cells. Deep cortex has T cells. Medulla, what does it have? It has both. So remember, superficial cortex, B cells. Deep cortex, T cells. Medulla, both of them. There also cortex contains dendritic cells. There are important features we need to look at here. There's something called medullary cords. Just know that it's present in the medulla. It's not like that significant. It's just projections from the cortex. It doesn't like, they didn't like give a significant relation to it. What about sinuses? Sinuses, lymphatic capillaries. They like dense together, and you have around them reticular fibers with macrophages. Yani it's also for immunity. We have sinuses in the, you see subscapular sinus in the cortex, and you also have in the medulla. So it's significant for lymph nodes. يعني مثلا if like directly which organ has sinuses, lymph sinuses, lymph nodes. So there are important features like for each organ, you need to remember them. Okay? Yalla, here's a question. Don't panic from lots of text, just read it. Okay, I have one. Yalla, do you have any answer? B. B, okay, we have a B, we have a C. B? B, طيب هلا at the beginning it's a case scenario. You look at the last sentence, it's literally the question. اللي هو what is the primary function of lymphoid tissue in the lymph nodes? Produce hormones? No, it's not endocrine. And filter out bacteria from the bloodstream and prevent their harmful action. To house lymphocytes and provide a site for their activation, or to produce erythrocytes. Now D is not, definitely not the answer. It's between B and C. Now both of them are like correct to an extent because we're trying for the immune function, but the right answer here that's like very direct, it's C. Because lymphoid tissue is for lymphocytes. It has lymphocytes and like for them to like proliferate, to meet antigens, to attack, to protect, and all of that. So the correct answer here is C. It doesn't have like the bloodstream. It can, because it cleanses the lymph. And it attacks pathogens. So it's not now very direct related to bloodstream. Now we're talking about the lymphoid tissue of the lymph nodes. So we care about the lymphatic capillaries and the lymph in it. So the correct answer here is C. Okay? What causes lymph to flow slower through the lymph nodes? A? Yes. Many afferent vessels entering, and we have only few vessels exiting, so it slows down because we need it to slow it down to cleanse it and kill the pathogens on it. Which parts of the lymph node show increased activity when antibody production is high? Just mentioned it. Okay. D? D? Yes. Germinal centers and follicles. Here you have outer follicles and you have germinal centers. Why is it not B? Because germinal centers are specific for B cells. يعني follicle has germinal centers, but it's not only germinal centers. Germinal centers is the one specific for B cells, which is antibody production is high. So we finished one lymphoid organ. We have in lymph nodes we have capsule, trabeculi, cortex, and medulla. Outer cortex, what does it have? The superficial B cells, اللي هن الfollicles, الgerminal centers. Deep cortex, T cells. Medulla, both. And we have something called medullary cords, and we have the sinuses. Spleen, we have capsule, trabecula also. The spleen is the largest lymphoid organ. There's something like special about it, that it has both functions, immune and blood cleansing. Lymph nodes is to cleanse lymph. Spleen is to cleanse blood from worn out blood cells. So we have in the spleen, you'll find white areas called white pulp, and you have red areas called red pulp. From the name, white pulp is which part? The lymphoid tissue for protection, immune. And the red pulp is for the blood cleansing functions, blood capillaries and all, and they're surrounded by macrophages to kill the pathogens in the, to kill the worn out blood cells. Kaman, it stores iron, it stores platelets, it stores monocytes. So in the book you'll see 3 functions. They're important to know. Storage, store platelets and monocytes. It also stores iron, kills worn out blood cells, and it can it produces erythrocytes in the fetus. So here we have in the important anatomical feature is that in the red pulp we have splenic cords. شو هدول? We have in the here if you look هلا طبعا this is what is this? White area, white pulp. The rest of it is red pulp. هلا in the red pulp شايفين هون in the something it's dividing. We have what are sinusoids? They're islands of blood. That's the translation of it, literal definition. It's islands of blood. So you have the red pulp. It separates these islands, and between them you'll see this is called the splenic. These are the splenic sinusoids, the islands of blood, and they're separated by splenic cords. Just remember them by name. Sinusoids. Where do you have sinusoids? Spleen. Okay. يلا هي homeostatic imbalance. D. What do you think? D? Yes, it's D. The spleen can repair itself. We found out. Alhamdulillah, it can replace itself. No need to remove it or transplant or anything. Liver and bone marrow can take its function. This is in case of splenectomy. If you remove the spleen, liver and bone marrow takes its function. But we don't want to remove it. It can repair itself. So this is it can important feature to know about it. هلا we enter malt, mucosa associated lymphoid tissue. So okay. Should I? B. Have one? B. Yes, okay. Yes, it's B. You have Peyer's patches. Where are they found? Small intestine. Which part? Important to know. They can bring initial part of the small intestine, middle part of the small intestine, last part of the small intestine. Middle. No, it's in the distal. Distal last part, distal portion of the small intestine. Thymus, obviously it's here, and it's a primary lymphoid organ for maturation of T cells. Tonsils, they are also malt. We'll talk about them. Appendix follicles. اللي هي appendix. Appendix in which part of large intestine? First part of large intestine. فعندكم Peyer's patches, last part of small intestine, and appendix, first part of large intestine. And it's very common to remove the appendix. I don't know if anyone here removed it. Okay, alhamdulillah. Okay, what do you think? You think D. B. We have B and D. B. Okay, so it's between B and D. فهو يعني it's always because both of them start with P and I also get them confused sometimes. هلا tubal tonsils. Tubal, where are they? Tubal is always related to, like it's related to the ear. You have the auditory tube, يعني inside your mouth there is a tube connecting to the ear. Around it there are tonsils, so that when you're eating, bacteria mashallah will reach like the ear very easily. So now we have tonsils around the tube to protect it. Pharyngeal lingual tonsils, EMS, this is like where EMS is definitely useful here. Lingual for base of tongue. So pharyngeal هلا it's at the end of the oral cavity, يعني when you eat. No, palatine tonsils are at the end of the oral cavity. Pharyngeal is like behind the nose, nasopharynx. The right answer here is pharyngeal tonsils, B. هلا palatine it's the largest. You have two palatine tonsils. They're the largest end of oral cavity, as I said. There's something special about them, is that they're most infected. Yes, they get infected and they also get removed. Many people remove them in tonsillectomy, but adenoids, it's special for pharyngeal tonsils, and this is like very important. It can easily come. What are adenoids? It's the pharyngeal tonsils, so don't get them confused. Okay. هلا MALT, mucosa associated lymphoid tissue, اللي هي tonsils, Peyer's patches, appendix, they're all what? Lymphoid follicles. They also have what? Germinal centers. They also contain B cells, these parts. So they're follicular. They also have some diffuse lymphoid tissue, but they all contain follicles. What's special about tonsils? Wait, I'm saying follicles, that doesn't mean they don't contain T cells. They contain both. But remember, they have follicles. There's something important about tonsils, that they have something called tonsillar crypts. Before this, are tonsils capsulated? No. They mentioned it's incompletely capsulated, so it's not capsulated. It's mentioned as no. Okay? So, and another feature is that it has crypts, the epithelium of the tonsil, it goes inward. So basically we're telling the microbe enter the tonsil, so we can kill it. So we're inviting it inside to kill it. It's a risk, but it's important function of it. We have tonsillar crypts. Last organ anatomy is the thymus. Peyer's patches appendix, we only need to know they have follicles with their location important. That's it. We don't there are not many details. Thymus, it's a primary lymphoid organ. Does it kill pathogens? No. Definitely not, because it's place of maturation of T cells. If an antigen comes, and what will happen? Premature activation of T cells. They're not mature yet. They're still undergoing the process of maturation, and so they don't have the knowledge to kill. So it's dangerous if they get antigens. That's why thymus has a very important. We have, remember, blood-brain barrier, because we want to protect the brain. We also have blood-thymus barrier. This is important for thymus because we don't want antigens to enter the thymus. So this is something important to remember about it. We have in it, it's two lobes, and the lobes are made of small lobules. Each lobule has cortex and medulla. Where did we see cortex and medulla? Lymph nodes. Lymph nodes has cortex and medulla. Spleen has red pulp, white pulp. So that's why I tell you relate the organs together. Cortex has rapidly dividing lymphocytes, few macrophages. Medulla has less lymphocytes, and cortex has more lymphocytes than the medulla. What's important about the medulla is that we have something, thymic corpuscles, keratinized epithelial cells. Strange, because we know keratin is on the skin, but we have keratinized epithelial cells in the medulla of the thymus. Why do we need them? For T regulatory cells. Hey, inshallah, when you pass this year, inshallah, enter year 1, you'll take next year the whole maturation process of the T cells and T regulatory and all of that. fun, a bit يعني overlapping, a bit complex, but it's nice, and you'll understand it more next year. But just remember, you have T regulatory cells in the thymic corpuscles. That's their place of development. Thymus has a special, remember spleen, what's special about it is that it repairs itself. What's special about thymus now?
C.
Okay. C. Okay, all of you see? Okay, hypertrophy in later life. هلا hypertrophy. Hyper يعني increase, more. Trophy, no, increase, we're increasing in size here. Does the thymus increase with age or does it shrink with age? It decreases with age, it shrinks. It's not C. لا, it's C, صح, except. Sorry, I didn't see the question. Yeah, I was saying why all of them are saying hypertrophy. Yes. All of the following, yeah, now we want the incorrect statement, which is C. No, yes, it decreases in life, so atrophy in later life, that's correct. Hormone secretion, remember? Endocrine, I put this is paragraphs from where? Endocrine chapter. It releases hormones, thymopoietin, thymosin, thymolene. I think they put it for you this year. You need to know they removed the thymus part for you. Okay, they put it for us last year. But just remember, it releases hormones. Providing immunocompetence, it's T cell maturation. Even if you don't know the B, of course it doesn't hypertrophy in later life. So you know this is it's C. Which lymphoid organ is located in the left upper quadrant of the abdomen? Left upper quadrant of the abdomen, right below the diaphragm. Where's the thymus? هون. طبعا مو in the abdomen. Where's the liver? Yes, so on the right side. Anyways, it's not, we didn't mention it as a lymphoid organ. Pancreas is somewhere in the middle. Spleen, yes, it's spleen. Spleen is right below the diaphragm. It's in the left upper quadrant. I cannot emphasize enough how important this table is. Even if you have like a few minutes, I don't encourage you to revise a few minutes before the exam, even though I do it myself. But if you just running out of time, just want to take the quick notes from this chapter, just look at this table. You have the features of all of them. You see lymph node, spleen, thymus, they all have capsule. Malt, of course, they're small lymphoid tissue here and there, they don't have capsule. The stroma, there's an exception here, is that all of them have reticular connective tissue, except thymus, it's epithelial. And remember, we have keratinized epithelial cells in it. What do we call them? Thymic corpuscles, اللي هن keratinized circular and they're placed for what? هي revision T regulatory cells. They're place of development of T regulatory cells. وهي the important features of each organ. So it's important. This table is very important. يلا, another question. Mass in the axillary region. It is not painful. Okay. A. Again, I cannot emphasize how much important this feature is. We also had it in year 1. By the way, we had a PBL case 2 weeks ago, and we were like, there's a mass, but it's not painful. When we thought, oh, there's a swelling, we thought, oh, it's lymphatics. But then it's not painful. We remembered this. We're like, most likely cancer, and actually it's tumor. So you feel like a doctor, you're able to diagnose. Because we have a mass, you'll think maybe lymphatic is not working or an infection or something, but because it's not painful, it's cancer. This is the homeostatic imbalance. I collected all the diseases for you, and it's useful to do it this way so that you just relate them together. Lymphangitis. Lymph. Angio is for what? What does angio mean? Vessels. Angio means vessels. Itis, inflammation. So the lymphatic vessels, they're supplied by vasovascular around them. when this gets, when an inflammation happens and all the vasovasin gets congested with the blood, and you'll have red streaks. So when you see red streaks here, you'll think it's a blood problem, but it's no, it's the lymphatic capillaries like getting congested. It's called lymphangitis. Lymphedema, we just said any block in the lymphatic system, edema will happen because we're not draining the excess plasma, that's the fluid. So it can be blocked by a tumor, and it will cause swelling by accumulation of fluid. Lymphadenopathy, we also call it swelling glands. Infection, we're fighting bacteria in the lymph nodes, so we're cleansing the lymph, but this bacteria can exit the lymph and infect the lymph node if it gets overwhelmed. And we have lymphadenopathy, we have swelling and pain. If it's cancer, no pain. So this is an important feature to remember. And lymphadenopathy, if the lymph nodes gets like pus-filled, it will also be called buboes. I don't think they like focus on this, but keep it in your mind in case. And for further, it always helps when you have a picture in your mind. That's how I used to do it when I used to see clinical conditions. What's this? Lymphangitis, red streaks, you see them. What's this? Lymphadenopathy, because it's the lymph nodes, it's swollen, and this will be painful. If it's not painful, cancer. This lymphedema. And I was like, what's the difference between lymphadenopathy and lymphedema? They're both swelling, but lymphedema to like everything will like get swelled. Lymphadenopathy will focus more on the lymph nodes, so it will be like this, balls. Okay. Yalla, last thing, developmental aspects. I always, the best news you can get is, oh, they removed it. Okay, I was just going to say the best news you get is that when they remove it. They removed it, alhamdulillah. Okay, so that's it, almost there, we're almost done with the year. Good luck. I know you'll, in the finals time, you'll be studying like this. But inshallah, you don't want to, you'll study so that you're not like this during the exam. You don't see the questions and you close your eyes, you'll be like this, inshallah. Okay? So you see and you know how to. Okay, thank you all. Thank you. I'll pass it now to my colleague, Noor.
Okay, hello everyone, I'm Noor. I think most of you already know me from the tissue. Okay. So most of you already know me from the previous tissues session. Today, me and my colleague Yasser will be presenting the immune chapter and hopefully making it more easier to understand, simplifying some hard concepts. And let's start with a question. Yeah. Can you tell me why? Yeah, can you tell me why?
Because it's systemic, it's not just one.
Yes, okay. Good job. Okay, so the immune system is divided into two, mainly the innate defense and the adaptive. We call the adaptive defense, another name for it is the third line of defense, and you have the innate defenses is divided into two. You have the surface barriers, which includes the skin, mucous membranes, you guys know this, and the internal defenses, which constitute of 5 components. Now, another name for the first line of defense is the outer innate, and also another name for the second line of defense is the inner innate. Simply includes like phagocytes, natural killer cells, inflammation, fever, antimicrobial proteins. Adaptive defenses is divided into two, cellular and humoral. Humoral is related to B cells and antibodies. Cellular is related to T lymphocytes and the cytotoxic ability of killing pathogens. The innate defense reduces the workload of the adaptive system. So how does it do that? Basically, you guys can imagine it as you have like the security, and then a little bit higher level is the police, and then you have the army. You can imagine it in the same way. So the security would be the first line of defense, and then police would be the second, and then army, so on and on. So it prevents the entry of the pathogens from the start, so it doesn't cause an infection later, and reduces the workload of the adaptive by basically doing that. So does a properly functioning immune system defend against transplantations and yes, okay. And in that case, what should we do when we do a yes, exactly. You guys are hearing the answers? Okay. Okay. Are we born with both innate and adaptive immunity? Which one? Innate. What about the antibodies that are from the mother? Are they enough to provide adaptive immunity? Why so? Can you?
Vaccines, infections to give adaptive.
Yes, exactly. Okay, this is a matching questions. By the way, they love matching questions in immune.
Second would be.
Yeah, you can raise your hand. Okay.
Second would be natural killer cells.
Okay.
Antiphagocytes.
Yes. Yes, that's right.
And then
Can I get another answer? Okay. Ahmad. Okay, what about cell-mediated immunity? Third, okay. And?
Gluten is first.
What?
Gluten is first.
Correct. Okay. The cells that are primarily responsible for immunity are? I want an answer من البنات. Yes, correct. Why not A? Why not the neutrophils? Okay, it's fine. Usually when we are talking about immunity in general, we mean the long immunity. So we do know that the neutrophils are usually for like acute infections, right? And macrophages are for the chronic. So we chose macrophages instead of neutrophils for this specific reason. Okay, what about this? Okay.
C.
C. Okay, why didn't you choose the second line?
Because first one depends on skin and mucous membrane. The second depends mostly on natural killers, phagocytes and stuff.
Yeah, so as you said, although like we have also antimicrobial proteins in the second line of defense, when they mention from the question itself, from the mucous membranes, they mean the first line of defense. And also in the first line, they say that they secrete antimicrobial peptides. You guys know the difference between peptides and proteins. Proteins are much larger, so they're related to the greater line of defense. Okay. Okay, guys, so whenever you see a table, it is important. Yani this summarizes all of the first section of the chapter, especially اللي هو functions and like what each layer secretes. So you can see here the acid mantle. Whenever you see acid, what is its main function? Kill bacteria, okay, and inhibits bacterial growth, okay. So you have the acid mantle of the skin, and this is a result of the skin secretions like sebum and sweat. All of these, they inhibit the bacterial growth. They also contain bactericidal chemicals. Bactericidal means it will kill the bacteria. Now keratin, it will provide resistance against acids, bases, bacterial enzymes, and also toxins. They also, I remember they got a question related to exotoxins. So exotoxin is basically a bacterial toxin. You guys are going to take later in year 1. But no, if they mention this, على طول remember keratin. Okay. Now for the intact mucous membranes. What are mucous membranes from tissues? Digestive, respiratory, and? What? They already said digestive. It's like the, yeah, urinary. Okay, I got you. Now it forms a mechanical barrier that prevents entry of the pathogen. Whenever you see mucus, it will trap the microorganisms. And it traps it in the respiratory and digestive tract. Why do you think so? Why in the digestive tract?
Very acidic.
Yes, that's right. So the acid in the stomach is going to kill the pathogen that is there. Now, the nasal hairs, basic information, it's going to trap the microorganisms. Cilia will propel anything like mucus related upwards, away from the respiratory, so it doesn't cause any blockage. Now for the gastric juices, this is related to why initially the mucus is sent to the digestive tract. And you have the acid mantle of the vagina. So again, acid inhibits the growth of bacteria, but in addition, because it's in the like female reproductive tract, it will also inhibit the growth of fungi. Now lacrimal secretions and tears, they contain an enzyme that you guys need to remember. It is lysozyme. It destroys the microorganisms. Also for urine, acid pH, again, I don't have to repeat it. inhibits bacterial growth. What is the difference between mucin and mucus?
Mucin mixed with water gives mucus.
Yes, that's right. I want from the girls. Yes? Yes, that's right. What is the enzyme name that would replace this and make it correct? Lysozyme, yes. Okay. They got us this question, by the way, and I got it wrong. It's not the exact wording, but you get the idea. Yes, correct. هلا you see options C and D, they're pretty unclear. حتى their words they're trying to confuse you. So always the answer sometimes is direct. You don't have to overthink it. Don't act smart. The answer is already clear there. Just choose it. And the acidity explains because again, it will kill the bacteria, and some bacteria in the mouth is helpful. So if you don't have these bacteria, you're gonna develop dental caries, and they're gonna cause this dry mouth and all of these symptoms. What's his name? What is your name? Naam? Zaid. Can you please answer this?
C?
Yes, that's right. Okay, I want you guys to get your phones out and scan this because we're gonna review the end with some questions. Okay, all in? Okay, I'm gonna start. Okay. Why did some choose lysozyme? I want to know who chose it. Why? It is an enzyme. They mean the antimicrobial peptides that we're gonna study now. Okay. So it is defensin. So defensin is basically a protein that whenever it is being released, it will be released by a neutrophil, it will pierce the membrane of this pathogen. So it will basically disrupt the microbial membrane. And it's not related to sequestering harmful molecules ولا إشي, it's not related to that. And for اللي هو stimulate the immune system, not yet, لأنه it's related to the second line of defense. Listen, it's too early to stimulate and alarm the immune system. Maybe you don't know what does dermacidin do, but you can guess sebum, acid. It's hunting. Okay. By the way, the top 5, I'm gonna give them ورق عنب. Okay? Okay.
Who's zebra?
How are surface barriers such as the skin and mucous membranes typically breached? This is easy. Okay. What happens after the surface barriers are breached and microorganisms invade deeper tissues? Remember? Police, army. A fever? Why fever?
Oh no, I chose version.
Who chose fever? Yes? Okay. خلاص مسامح. Okay. Which structural modification in the respiratory tract helps to prevent the invasion of pathogens into the lower respiratory? Who chose the blue one? Why so? Tell me your explanation.
The mucus which is secreted from the goblet cells.
Okay.
Yeah, the pathogen.
Yeah, but they mean. Bismillah, hold on. Yeah.
Cells is present in the epithelia of the upper respiratory.
Yeah, but mainly when they're talking about the respiratory system, we do know that we have like nasal hairs, we have اللي هو also شو كان اسمها cilia. All of these they try their best to keep the pathogen away from the lower respiratory tract.
But yeah, the cilia also moves the mucus.
Towards where? The mouth. So yeah, so it prevents from. Did you get it? Okay, I'll explain it. هلا أنت you have mucus coated hairs in the nose صح؟ These nasal hairs they prevent the pathogen from entering اللي هو respiratory tract, whether it's upper or lower. So from the beginning it's preventing the entry to the lower كمان at the same time. And even انت you're saying the mucus صح اللي هو by goblet cells. بس هون in the question they are asking for the structural modification. ما هو in other parts of the respiratory you don't have hairs. You don't have hairs in your like your larynx and all of that. You guys took respiratory ولا لسه؟
We're doing it.
Okay. This is true regarding the relationship between the skin, mucous membranes, and inflammation and the innate immunity. None of the above? And I put it عشان no one chooses it. Okay. Skin secrete defensin only when there is a bacterial infection. هلا defensin is always secreted, but it increases significantly when you have an infection. And it's not related to skin, it's related to phagocytes that release them, اللي هو neutrophils. Mucous membranes rely on adaptive immunity to control bacterial growth. No. What do mucous membranes rely on? We had the acid mantle. We also had what? We had mucus. We had all of these. هدول كلهم they tried their best to control اللي هو bacterial growth. Last question. Which function does the mucous membrane in the respiratory tract perform in the defense against pathogens? Okay. Good job, guys. Who's zebra? Mean zebra.
It's me.
Okay. Good job, Ahmad, and I think it's you, right? Who's Zain? Okay, good job. And then we have Ibrahim. Who's Ibrahim? Okay, and it's not showing the bio. Okay. Guys, خلاص you know yourselves. ارفعوا ايديكم انا بعطيكم. تعالوا. إبراهيم. أعطي عماد وأنت. Zebra and there's someone زيد ولا زين؟ زين.
زين اللي هو هذا.
Yeah. Thank you. Okay. Now the actual immune system. انسوا لي كل شيء قبل وهلا ركزوا لي. Non-specific cellular and chemical body defenses. هلا احنا we're starting with the second line of defense. Can you guys name for me what are the components of it? Second line.
Natural killers, parasites, macrophages.
Huh?
Macrophages.
Okay. Yeah, phagocytes. They're part of phagocytes. What else? Fever. And then? Inflammation. And last one. They said it. Antimicrobial proteins. هلا they're easy to remember, but I did make a mnemonic for you guys, which is frequent pain. You can use it to remember. And I want to highlight the most important part of the second line of defense is the inflammation. حتى you're going to see like so many slides about it. Inflammation is going to recruit white blood cells, macrophages, mast cells, and dozens of chemicals to the area so that they can be able to remove the pathogen that is there and repair. هلا recognition of pathogens. This is important to understand. We're going to take it more in year one. يعني don't be afraid من هاي الصورة. I just put it for the sole purpose of understanding. So if you're going to focus with me, here you see a dendritic cell. You guys know what is a dendritic cell? What is it? Yes. Why? So that it can present antigens to the other lymphocytes. The dendritic cell itself, it will have receptors. These receptors, we call them pattern recognition receptors. From the name itself, they're gonna, whenever they see a pathogen, they're gonna recognize a specific pattern. And once they do that, they're gonna engulf this pathogen, and then they're gonna take how can I say it? How you guys into have common ID, it identifies you the same way. They're going to present it on their surface, and they're going to present this antigen to the lymphocytes. The beginning of this process, and it needs first to bind to that pathogen, right? It will have these receptors, we call them pattern recognition receptors, and a subclass of them is toll-like receptors, okay? We have 11 types in humans. Each recognizes a specific class of microbe. You don't need to know them right now. You're going to take them year one, but not all 11. That's it. It's pretty simple. Yani they just bind to it and they recognize it first. This is an example in your book, which is that TLRs are found on various cells, macrophages, and it explains why, because macrophages are antigen-presenting cells. We also have on epithelial cells of the GI and respiratory tract. And how they work, I want you to imagine أنه هون أنا عندي antigen-presenting cell, and it has this toll-like receptor that is hanging out of it. And then we have this example in your book is for tuberculosis bacteria. So this glycolipid is like, يعني, it signals to the immune system and wallah there is a pathogen, you know, and whenever the immune system sees this glycolipid, بعرف إنه this is tuberculosis, and it kind of hints for itself. Once it does that, the TLR is going to bind to it, and then it's going to recognize and signal for the immune system that there's a pathogen going on, there's a disease. And the same way here happens. Here, this is a component of the negative, gram-negative bacteria, Salmonella, once the TLR binds to it. Events of phagocytosis. Can anyone name some phagocytes? Phagocytes. Neutrophils, macrophages. Then, okay, but these are part of antigen-presenting cells. Okay, so the main ones for the innate is the neutrophil and macrophages. What happens in the first adhere to this pathogen using the receptor, the same what I just explained, and then the phagocyte will form pseudopods. Anyone knows what are these? They're like extension, exactly. They kind of like come and grab it, and they're gonna eventually engulf this particle, forming something called a phagosome. And in each cell, we know there are lysosomes, and specifically the ones in neutrophils and these phagocytes, they have specific enzymes that are gonna kill this pathogen. A lysosome will later fuse with the phagosome, and it's gonna form something called phagolysosome. Okay. In the phagolysosome, you have toxic compounds, and these lysosomal enzymes are going to eventually destroy the pathogen. And whatever was not digested, yani residual stuff, they're going to be basically excreted by exocytosis. Role of phagocytes. When do they mainly act? They act when pathogens enter connective tissue beyond skin and mucosa. So basically when they breach the first line of defense. The most aggressive phagocytes are macrophages. Whenever you have an infection, if you want to know if it's acute or chronic, you look at the complete blood count. If you see the neutrophils are high, that means acute. If you see macrophages, that means chronic. Macrophages, they're derived from monocytes, and they exit blood and enter tissues. We have two types. And free macrophages, which basically roam around like random tissues, and we have the fixed ones, which reside in organs. يعني هما they don't leave this organ, they stay in it, وحتى they have like a whole complete different name. Sometimes phagocytosis, the whole process itself, fails, and especially in tuberculosis. You guys know what is tuberculosis in Arabic? مرض السل. سمعتوا عنه قبل؟ السل. حدا سمع عنه قبل؟ سمعتوا؟ Okay, no need. الله يبعدكم عنه. It basically resists lysosomal enzymes, and it's gonna survive inside this phagolysosome. يعني النيوتروفيل ما لها فايدة. لأنه يعني هي حبسها, yes, it traps it, but still whenever the immune system is going to be compromised, يعني a person has another infection, this tuberculosis gets activated again and acts against the body. So what happens, we need the help of the aggressive phagocyte, which is the macrophage, in which helper T cell, it's part of the adaptive. Helper T cell is going to activate this macrophage by releasing specific chemicals, okay? And once the macrophage is activated, it will cause something called a respiratory burst. Inside the macrophage itself, it has so many lysosomes, and it's going to release the free radicals and اللي هم oxidizing chemicals. All of these, they're like bleach. يعني تخيل تمسك bleach and like you throw it on someone, you just burn it. In the same way, I did repeat so many times that acidic pH inhibits bacterial growth, right? But what happens here إنه البacteria is very resistant. It is very كيف بيحكوا عنيدة. So what happens, we do the opposite. So instead of like lowering the pH and making it acidic, we're gonna raise it so that we can kill this bacteria and the osmolarity and the phagolysosome. And in this way, we activate the protein digesting enzymes and we kill it. هلا neutrophils, it has another way of killing pathogens, except of like يعني instead of just eating it, it can release defensin اللي هو حكيت لكم عليهم. It pierces the pathogen membrane. You're gonna ask me a good question. Why don't we use defensin from the start for TB وخلاص يعني من البداية? Defensin is not enough, and you're gonna take later كمان in year 1 a bit about bacteria. So like tuberculosis is a very like strong bacteria. Just defensin is not enough for it, so we need the help of the macrophage. We got this in our exam. Yes, that's right. Okay, why not C?
It's because we couldn't engulf them, that's why we couldn't actually kill them or reach the point of using defenses or respiratory.
Exactly, yes. Do you guys get it? Everyone gets it? If you don't get it, please raise your hand and tell me عادي anytime. Yeah, tell me.
I don't get why it's not C.
Why it's not C? هلا in your book it's mentioned إنه neutrophils can also do respiratory burst, right? If I'm not mistaken, is it? Yeah. It's just not enough. يعني it's not about كمان idea of not enough. هو if it did من البداية respiratory burst, it's gonna kill this bacteria. But the question is asking you what is like the problem of this process that caused إنه البكتيريا to not die. And I don't have to أحكي لكم إياه بالعربي إنه يعني النيوتروفيل it can do two processes. One of these processes fails. That's why we use respiratory burst. So it's asking which of these processes has failed. Does it make sense? Sometimes the question is very clear. It's just the wording that they play with. Okay. هلا we have challenges بالphagocytosis. I did mention بالexample الglycolipids على tuberculosis bacteria and how it signals يعني the immune system can recognize it and know it is في infection going on. Sometimes the bacteria, they're very smart. With an external capsule, they can hide the carbohydrate components, and the immune system basically cannot recognize them. Even if they have اللي هو toll like receptors على dendritic وكلها, they still can't recognize it. So we have a process called opsonization, which basically we use complement proteins and antibodies to code this pathogen. I remember when I was foundation, Dr. Abu Bakr gave us an example of like a donut. If you see a plain donut and another one that has like all of this chocolate and sprinkles and all of these stuff, which one would you choose? The sprinkled one, not the plain one. So the same way what happens in the immune system, opsonins basically coat this pathogen so they make it more delicious for the immune system so that it can eat it. They greatly accelerate phagocytosis. By the way, I forgot to mention some questions I didn't explain them yet. يعني their introduction to the topic. So this is one of these questions. What?
C.
Correct. Do you guys agree that it's C? Do you agree? Okay. Hela, there's another problem, but we have solutions this time. Sometimes ingestion fails due to the large size of the pathogen. In this case, we release some chemicals with extracellular fluid to kill it. And because of that question, you did see that the neutrophil kills itself by mistake when it's killing the pathogen after that. So for this, we have the macrophage that replaces it, and it can survive longer and continue the killing. Which cells play a vital role in the immune system by assisting lymphocytes and are derived from monocytes? Exactly.
I have a question.
Yes.
So the neutrophils die when they attack a large pathogen.
No, it's like two separate situations. Sometimes the pathogen is too big, so in this case we release chemicals to kill it. And sometimes, not sometimes, this is always, neutrophil, when it kills a pathogen, it dies. Yani it's like self-suicidal. Yani I kill a pathogen and I also die. That's why it's only present in acute inflammation. Does it make sense? Yes. A macrophage lives longer. Yes? D, that's right. Okay, natural killer cells, they are basically the police in the body, and whenever you see natural killer cells, I want you to remember two stuff: cancer and virus infected cells. They always kill it before the adaptive immunity kicks in. هلا you guys took in blood chapter اللي هو A granular lymphocytes, right? So natural killer cells are lymphocytes, but they are granular, okay? And they are not that picky. They kill, like, they detect general abnormalities, which is, one of them is the lack of MHC. I'm going to talk about it in a few slides. They're not phagocytic, and the way that they kill is whenever, like, they see a pathogen, they come in close contact with it, and they induce apoptosis. Apoptosis is basically programmed cell death. Yani it tells it, go kill yourself. in another way. So the same way that natural killer cells kill, cytotoxic T cells also kill. They are in the adaptive. One of my doctors in year 1, you guys are gonna see her, Dr. Noor, she says that natural killer cells and cytotoxic, they're sisters. It just differs in which part of the immune system. Natural killer in innate, cytotoxic is an adaptive. Okay, so what are causes of inflammation? Ali صوتك. Infection, okay.
Trauma, chemicals.
Yes, okay, and?
Dermatitis.
Heat. Heat, yes, okay. Okay, so what are benefits of inflammation? Okay, prevents spread of infection, clears debris, and helps the tissue to repair. Yani the tissue cannot repair while there's still an infection. And it also alerts the adaptive immune system. What are the cardinal signs?
Heat.
Yes? Heat, redness, swelling,
And immobility.
immobility. هلا you guys would think إنه immobility معناها إنه يعني this is something bad. بس هو بالعكس, when it is immobile, يعني the joints, especially the area that has been exposed to trauma, and this way it will let the area to give it time يعني to repair. So it looks like a disadvantage at first, but it's actually يعني for the repair process. هلا inflammatory chemical release. You guys focus for this because this is important, and I need to stay here, but no, focus on the left side. It begins with a chemical alarm, which basically the presence of inflammatory chemicals, all of these in the extracellular fluid. هلا, can you guys give me sources of inflammatory chemical? And what are some sources?
The damaged epithelial cells.
Yes. What else? Zain, was there another source? It's easy. Yes, so the immune cells. And one of the examples in your book is mast cells when they release histamine. Other chemicals that we have include kinin. Can anyone name another one? Prostaglandin. What else? Cytokines, correct. Okay. What if the inflammation is not caused by any of these like sources and it is caused by a pathogen? Complement, okay, and it's going to trigger the release of inflammatory chemicals. The effect of inflammatory chemicals, you guys took it from blood vessels, dilate the arterioles, make capillaries leakier. What else? Attract phagocytes to the area because they increase blood flow. Yes, pain, we're going to get into that. Oh, yes, this is a question. Yes. You guys agree? Cytokines, yes. The sneezing, runny nose, and okay. Okay, vasodilation and increased vascular permeability. This is pretty easy. Whenever you remember vasodilation, increased blood flow, arterioles, they dilate, leaky capillaries, yes. Hyperemia. Hyperemia will give you two signs of inflammation, which are redness and the heat. Increased capillaries increased permeability of capillaries and venules will allow exudate to seep into the tissues. What is an exudate?
Fluid buildup.
Yeah, what does it contain? Proteins. Yeah, so it contains clotting factors and antibodies. Okay, so I cannot stress enough how important tables are. Yani if you, again, like how my colleague Israa said, whenever you see a table, it's like a summary of the topic. And this is very important. So I don't want to waste more time on it. You guys can review it later. But this is very important. اللي هو البروستاجلاندين. It induces اللي هو الpain. It also induces اللي هو neutrophil chemotaxis. This is you're gonna take it pathology in year 1. There are many prostaglandins. There are a lot. But yeah, that's it. I already mentioned this. Functions of exudates sweep foreign material into lymphatic vessels. Why do we need clotting factors? Why do we need a fibrin mesh in the area?
So that the pathogen doesn't go and spread.
Yes, okay. And we can also assume that after we get rid of this pathogen, if there is any like injury, we can also repair it. We can use this fibrin mesh for it. Some bacteria like Streptococcus, they produce enzymes to break down the clots, helping them invade surrounding tissues. I said this so many times. What do you think? See? Okay. What does edema cause? B. Why? Why does it cause pain?
Nerve inflammation causes swelling and pressure
On nerve endings. Exactly. Okay, so pain can be caused by the pressing on nerve endings, but it also can be caused by bacterial toxins and by the release of prostaglandin during the inflammation process. We have aspirin. It will basically relieve the pain by inhibiting the prostaglandin synthesis, because we did mention that prostaglandin causes this pain. Soon after inflammation begins, makes sense why it arrives first in neutrophils. This is important. They got us a matching question about it. اللي هو الphagocyte mobilization. So leukocytosis first starts off with in the area that is inflamed, we have leukocytosis inducing factors. These are gonna induce اللي هو neutrophils to exit the red bone marrow and sweep into the blood. And then after that, once it gets close to the area, the capillaries, they have like on the endothelial cells from the inside, we have something called cell adhesion molecules. Cell adhesion molecules are gonna basically allow the neutrophil to stop, slow down, and roll on this endothelium, and it will clinch to it. Then we have the other process, which is diapedesis, which is basically the squeezing of the neutrophils through each endothelial cell into the area. And now once we're finally in the tissues, we have اللي هو chemotactic. You guys know what are chemotactic factors? Chemotactic chemicals?
The chemicals that allow the immune cells to follow.
Yes, it's like a trail for them to follow along. So chemotaxis happens in here, neutrophils follow the chemical trail. What if I have a problem with margination? Oh no, حكيت الإجابة بدل السؤال. Okay, what if I have a problem with اللي هو the production of cell adhesion molecules? It would be margination. If I have a problem with leukocytosis inducing factors, leukocytosis. Okay. Arrival of monocytes, transition to macrophage. هختصر لكم. Basically, monocytes, they are weak phagocytes. So what happens when there's an infection and they need to go to these tissues, they're going to swell, they're going to develop so many lysosomes, and after that they're going to become macrophages, and they're going to replace these neutrophils. And they dominate the chronic inflammation. صح, correct. Okay guys, bio 2, homeostatic imbalances are the most important thing. إذا بدكم تراجعوا شيء قبل الامتحان دايما راجعوهم. Yellow pus formation is a result of collection of debris, of also fibrin mesh, dead pathogens, all of that, they accumulate in the wound. The problem is sometimes we fail to clear this debris, which is going to form اللي هو abscess. Okay, and this abscess needs surgical drainage. And if you don't drain it, it's going to spread to other parts of the body and even can cause sepsis. Do you know what is sepsis? يعني the whole blood كله بيكون فيه الباثوجين. هلا because we did mention tuberculosis, resist digestion, and all of this, we can prescribe antibiotics. And in such cases, granulomas form. A granuloma, يعني, excuse my drawing, but what happens when you have the infected macrophage and the bacteria is trapped there, they're not able to kill it. So another uninfected macrophage surrounds it, and we also have an outer fibrous capsule. So this is what is a granuloma. And this is the same idea.
When the immune system is ever compromised, the bacteria may get reactivated and it will break free, leading to clinical disease symptoms. So this part of the recording was cut. I'm going to summarize it in short. So a tissue injury is going to release some inflammatory chemicals that we have talked about in the other figure. That is going to allow the arterioles to dilate, which will cause local hyperemia, which will have increased blood flow to the area. And because of this hyperemia, we'll get two signs of inflammation, which is redness and heat. Now, this heat is going to increase the temperature in the area, and this increase in temperature will increase the metabolic rate of the cells, which will speed up the healing. Now, for the increased capillary permeability, it will make the capillaries leak the fluid, exudate formation, and this protein-rich fluid is going to leak and cause the swelling. This swelling will press on the nerve endings, which will cause the pain. And because of this pain, you're not going to be able to move your hand or your leg, the area that is affected, يعني. And this will give the area time to heal, and you're basically resting it. So this will also speed up the process of healing. Now, for the اللي هو leaked clotting proteins form interstitial clots. All of these, they're trying to basically surround the pathogen so that they can prevent its movement to the other areas of the body. And this will speed up the healing process, and then attract neutrophils. We have discussed this before, which is the phagocyte mobilization. Now here are some questions. As part of the inflammatory process, the following occur except. Now, capillary permeability, yes, it does increase, and proteins leak into the interstitial fluid. And when cells are injured, they do release chemicals. Now, pain receptors are activated by the inflammatory chemicals due to the swelling. Now, eosinophils and platelets are attracted to the area of inflammation. No, eosinophils are not. So that is the answer. The redness and the heat are due to a local effect caused by vasodilation. Now, antimicrobial proteins. I do know that complement proteins are completely excluded from your material, but I know that interferons are included. They basically help to enhance innate defenses by attacking the microorganism or inhibiting their reproduction. Now, it will inhibit their reproduction indirectly, which is explained by interferons. Now, interferons are basically proteins that are released by infected viral cells so that they can protect the uninfected cells. Now, this whole process and figure is also excluded, but it's good to know how it works so that you can understand it, but I'm not gonna go in details since it's excluded. Now, key features of interferons, they're made in response to one virus, and it can protect against others as well. Each type of interferon has slightly different effects. You can notice that interferon gamma is released by lymphocytes and it activates macrophages, while interferon alpha and beta, they're released by most body cells, and they have this antiviral effect. Now, I did mention before that natural killer cells, they kill virus-infected cells and also cancer cells. So if you want to think about it also in another way, if interferon alpha and beta, they're going to activate natural killer cell, and this way they also kill cancer indirectly. So interferon effect, here you can even see the clinical use of interferon, that fights cancer indirectly by activating both macrophages and natural killer cell. Now, for the natural killer, interferon alpha and beta. For the macrophages, it is gamma. And it is also used to treat hepatitis C, genital warts, and multiple sclerosis. Now, interferon only affects infected cells? No. Actually, the infected cells, they cannot help themselves, so they want to help the other uninfected cells. Is it specific for individual viruses? Not really, because it's part of the innate. Again, we're still in the non-specific part of the immune system. Induce enzyme synthesis in the target cells. Yes, that's the correct answer. A cancer patient receiving consultation asked why they were prescribed interferons for treatment, since they believed interferons were typically used for viral infections. What is the best explanation you can give? In here, the answer is A. It is direct. All of the other answers are trying to confuse you and they're not related in any way. Yani, example C, this option. Dissolved cancer cell membranes in process similar to antiviral defense? No. We didn't even take this in much details. So, yani, that's not the answer. Suppressed tumor metabolism? Not really. It kills cancer. Assured a patient that moderate fever is helpful. Which of the following would be explanation for that? Now, moderate fever, and in your book directly mentioned that once your body temperature increases, it will signal for the liver so that it can basically gather and trap all of the iron and zinc and kind of steal it from the bacteria so that the bacteria can die, and it will inhibit the bacterial multiplication because the bacterial multiplication does need iron and zinc. Now, fever, it's a systemic response to infection. Pyrogens, this is an important keyword. It is released by leukocytes and macrophages. It acts on the hypothalamus, which raises body temperature. I already said this information, it is important, and it also increases metabolic rate, which speeds up the healing process. Now, this table is also very important. Try your best to memorize and understand every single part of it. And here's a matching question. Please pause the video if you want to give yourself some time. And the answer is A. Now, adaptive defenses, these are the key characteristics. So specific systemic memory involves B and T lymphocytes. Whenever I say B cells, I mean the humoral immunity. I also mean the antibodies. And the antibodies, they work against extracellular targets like bacteria, toxins, and even some viruses. Now, viruses mostly the way that they work is that they go inside the cell and then infect it. In this case, we use cellular immunity. But if there are extracellular viruses that can survive without it, without going into a cell يعني, and reproduces and all of that, we would use اللي هو الhumoral immunity. هلا الcellular immunity, it kills cancer cells, foreign graft, infected cells. It works directly either by like cytotoxic activity or indirectly by releasing chemicals to activate other immune. An antigen is basically any substance that triggers the adaptive immune response. It's a foreign substance, and the complete antigen has two components, reactivity and immunogenicity. Now, reactivity is basically the ability to bind to antibodies and lymphocytes, while immunogenicity is the ability to stimulate the lymphocytes proliferation. Hapten, it's not a complete antigen. It's a small molecule. It can be example like penicillin. Penicillin is an antibiotic. Now, hapten is not immunogenic, but it is reactive in a sense that it can bind to lymphocytes and the antibodies, but it will not stimulate the lymphocyte proliferation. Though, if the hapten goes and binds to a body protein, like a protein in our body, it will cause an immune reaction. It will stimulate the lymphocyte proliferation. So it will be immunogenic in the case if it binds to a self protein. Now, antigenic determinants, basically, you can see here on the picture on the right, you can see an antigen. Now, on the surface of it, it has many antigenic determinants. And in this case, it explains why we can have multiple lymphocytes all acting on one antigen, because on the surface itself, we have many different, زي lock and key. So we have many different keys, and here comes the lock. يعني لكم تقريبا antibodies in this case. Now, which of the following is characteristic of complete antigens? It's reactivity with an antibody. Okay. A 41-year-old manager is admitted to the accident and emergency department. Assessment of her condition reveals that she develops wound infection. The doctor decides to start her on penicillin. Notice the keyword. Within 20 minutes, her face begins to swell and she's wheezing. Her tongue is swollen and she's unable to open her eyes because of swollen eyelids. On examination, she looks anxious, flushed, and short of breath. She was diagnosed as an allergy case. Her vital signs are summarized here in the table. Now, this is not the exact one, but it's similar. We can notice that her blood pressure has dropped, hypotension, tachypnea, tachycardia as well. Her body is reacting, it's like an allergic reaction in here. So in this case, the answer would be hapten that binds to self-proteins, because in this case, it will be immunogenic. Now, the same question, but this time it says the sneezing, runny nose, and watery eyes make a person having allergies feel so miserable as a result of histamine. histamine released by mast cells and basophils.
Okay, MHC, this is important. I know in your book it's like only one paragraph, but MHC is basically a protein that is on your cells in your body. So how can I know this is a cell that is from my body point of foreign? By recognizing the MHC. It is found in all body cells except RBC, and they vary from person to person. So the reason why sometimes we have a transfusion reactions they feel and the transplants is because the MHC basically differs from person to person. There's also another point in your book that MHC molecules, they present antigens to T cells. You're gonna think of it and this doesn't make sense. احنا حكينا ان MHC it is on my cells, and in this case they are saying it presents antigens to T cells, صح? It doesn't make sense. In this way they're saying it's like for the pathogen. PACademia, you don't need to know MHC 1 or 2, but for you to understand. And you're gonna feel like few contradictions in the book, and this happens a lot. In MHC molecule 2, it will present antigens because it is on the dendritic cells, human antigen presenting cells. So when it engulfs the pathogen and it's going to represent part of it that basically identifies that this is a pathogen on the MHC, it will help it so that it can present to the T cells. That's all you need to know. And this is a table that basically simplifies everything from chat, GPT, that simplifies. And adaptive is divided cellular and divided immunity. These cells we have 3 types. You guys need to know the liquid names, by the way. CD8, CD4, ADD. They don't have to mention cytotoxic. You just need to know the other ones. You also have regulatory suppressor T cell, which basically inhibits B and T cell activity. And sometimes we have autoimmune disorders and all of this. What happens is that basically your regulatory suppressor T cell, it doesn't work the way that it is supposed to. So that's why it causes an autoimmune reaction. For the B cells, it produces plasma cells, and these plasma cells, they produce an antibody. If I tell you a child is born with a congenital disease that results in total loss of B cells, what would be a consequence?
No protection.
And antigen representing cells, we have 3. And then B lymphocytes, macrophages, and dendritic cells. Dendritic cells being the most powerful, or not powerful, but not the most common antigen present. Remember, it's the sister of natural killer. And notice the keyword invaded body cell. Invaded body cell معناها it's a hence for the virus. Okay. So this figure is very important. سيبكم من كل هالكلام, just focus on الكلام اللي بالfigure itself. هلا B and T lymphocytes, they originate in the red bone marrow. A maturation site, which basically constitutes of your immunocompetence and self-tolerance. Both of these, their location differ. The B lymphocytes, they stay in the bone marrow. But the T lymphocytes, they go to the thymus. You guys took the thymus, صح? Immunocompetence and self-tolerance. Self-tolerance basically is when the T lymphocyte or the B lymphocyte, it will see a self-antigen, it will not react against it. And this also prevents autoimmunity, so it doesn't attack my own body cells. Immunocompetence is that each lymphocyte it can recognize one specific antigen. These receptors that I'm talking about are specific for lymphocyte types, T cell receptor, B cell receptor. And they're important because they only recognize one pathogen. Use this. Thank you. Okay. Maturation occurs in the primary lymphoid organs, and this is all you need to know. Significance of the appearance of antigen-specific receptors on lymphocytes. B. D, to recognize, yeah, only a one specific antigen, yes. Okay, now seeding secondary lymphoid organs. Can anyone name some secondary lymphoid organs? Lymph node, spleen, yeah, okay. So what happens, yes, you have mature T cells and B cells, but they're naive. Naive يعني basically they're not yet exposed to antigen. They're mature in a way إنه خلاص they know how to like they tolerate اللي هو self antigens and all of that, but they're still not exposed to antigen. So what happens إنه they go to the secondary lymphoid organs and eventually they're gonna encounter the antigen finally يلي that was brought by مين؟ الانتيجين. No? Exactly. So this entering into tissues is controlled by homing signals اللي هو الكام cell adhesion molecules the same way. هلا antigen encounter and activation, this occurs in the lymph node or the spleen. This is related to this يعني if this doesn't happen ما في antigen encountering. Lymph nodes are well positioned to catch antigens because you know the lymph flows there and all of that. هلا when a naive lymphocyte binds to a specific antigen, it will basically be selected for activation, which is clonal selection. Also Yasser is going to talk more about this. هلا if proper signals are there, we're going to have proliferation and differentiation. From the word itself, proliferation, what does it mean? Making copies of it more. And differentiation, they will differentiate into like memory cells, into اللي هو effector cells like cytotoxic, which are directly going to attack. Yeah. B? Yeah, okay. Yes. What about this? Also A? Clonal selection, حرام عليك. Yes. Okay. A patient came into the clinic with swollen joints in the synovial membrane. It appeared that this was an autoimmune disease caused by the patient's own lymphocytes attacking self-antigens. صح. Matching question. Do you agree? She's still reading. B. Which cells are responsible for engulfing antigens and killing the APC? They're obvious. And we did mention the types. هلا the idea that you mentioned إنه macrophage can basically activate T cells or something like that. The most important and efficient APC for T cell activation هو dendritic cells. And then comes the macrophages, which basically activates the effector T cells. And what happens, I think in your book it's mentioned T helper cell, صح? Yeah. هلا المacrophage when it activates a T helper cell itself can help activate this macrophage to become an aggressive phagocyte. فيعني if the infection, like what happens in granuloma and all of these, if it's not able to get rid of it, that's what the T helper cell is going to do. هلا for B lymphocytes, is it here? Yeah. What happens in activated B lymphocytes, they can activate the helper T cell. And in the same way, helper T cell can activate other naive B lymphocytes. So يعني the B lymphocytes can activate itself indirectly through the T helper cell. It might seem a little confusing, but you can see in the T helper cell is kind of in everything. That's why it's important to both the cellular and humoral. Then in humoral, B lymphocytes antibodies we're talking wise. T helper cell, when it's activated by the macrophage, it can activate other part of the cellular immunity. This table is important. Oh, yeah, that's it. Cell-mediated immunity would be least effective against D. D. What? صح. You guys have like 3 questions of Sausan coming to the clinic, get ready for it. B, صح. Why not cancer? Yes, and cancer is hard. It's not tender, like you mentioned, and it's not painful. How Sausan's nodes become swollen? Okay, more related to the idea in pathogens when they're trapped, they can cause this inflammation because if they're large in size, they're going to cause the lymph nodes to become even more swollen than a normal infection. The following blood cell types are expected to be high in number. And also remember, she came because of a recent infection. B, correct. The doctor decided not to give a drug to decrease her body temperature. Why? A. No, it's not A. Fever will increase the metabolic rate of the tissues. What happens of the tissues? So on. Of the tissues to accelerate the healing process. So it's not related to that. It's mainly related to iron, and the zinc is trapped in the liver and spleen. Okay, this is a matching question from your book. By the way, we're almost done. Yeah, this is it. Okay, but there's another option. Safe. No, it's not C. Dendritic cell is not a phagocyte, yani. B and G. Okay, what about 2? Release histamine. Mast cell and basophils. This is in your blood chapter. What about release porphyrin? I didn't discuss it, but porphyrin is basically natural killer. Okay, and you know it, right? Cytotoxic T cell. Porphyrin is basically protein that goes on the pathogen membrane, بيجي حواليه, and it forms a hole, and from this hole, whatever fluid is in the extracellular part is going to enter this pathogen, and it's going to lyse the pathogen. What about lymphocytes? A, E, F. F, right? And H, helper T cell. What about natural killer? It's a lymphocyte, but it's a granular one. Granular. Okay, effector cells of the adaptive immunity. صح? Yes, correct. Yeah, صح. Antigen presenting cells? Yes. 2 more. No. G and F. Yeah, macrophages, B lymphocytes, and dendritic. Okay. And that's it for my part.
The chart. What is the name of the cell? Exactly, plasma cells. And a small portion of them will remain as memory cells. And this is important because in future infections, this is what will help your body immediately recognize the pathogen. So let's just keep that in mind. And let's move on to the next question. So B cells respond to initial antigen challenge by doing what? Forming a large number of cells that are unlike the original B cell, reducing its size, producing progeny cells that include, Correct, yes, absolutely. So why is, for example, D not correct? Why is D not correct?
Because first time they take 3 to 6 days to form.
Exactly, correct. So the keyword here is initial antigen challenge, and it doesn't react immediately by producing antigen-specific antibodies. It does that with a time lag, and we'll see that as well in a bit, right? And this actually ties into what I'm going to be explaining next, which is immunological memory and what provides it. So what is immunological memory?
Memory is remembering the infection that happened.
Exactly. So they are remembering what's happening, and they're using this knowledge against the antigen in the future to prevent it from causing the same harm that it caused the first time, right? And who is responsible for this? Who provides it? Memory cells. All right. So what are the cells in your body that have memory cells? Or specifically what lymphocytes or what immune cells? B and T, exactly. Both B and T have memory cells, and this is very important. And you'll know that next year. So keep that in mind for now. And forget it during the summer and come back to it next year. All right, so that's immunological memory, and the B cells and the T cells provide it. And let's see how this immunological memory works. So this is the same isolated black-colored memory cell that I found in the original slide. And the whole idea of immunological memory is that when obviously memory cells flow in the bloodstream, so when it binds to the same pathogen again, I use the same one, what happens is insane and huge proliferation of the memory cell to produce a lot of plasma cells, and obviously there are going to be some memory cells, because if you produce all plasma cells, and obviously we know that plasma cells have a limited half-life, so they're going to die eventually, and when they all die, you lose the memory, you lose the immunological memory. So you produce a specific fraction of memory cells within each exposure to the antigen to make sure that you never lose this immunological memory. So this is how I like to visualize it, is that there's insane proliferation. So now it's important to remember it's important to know the differences between when you expose to the antigen the first time and the second time. So which exposure is the slowest exposure for the immune system and why? Exactly, why is it?
Because it takes time to build up the memory B cells and plasma cells.
Yes. Okay. So there's a multitude of reasons of why immunological memory, or specifically the first antigen exposure, takes time. And when you look at the whole process of the first immune response, you'll see that there are some points where the body is lacking behind. The body is not doing its stuff the most efficiently. And that's, for example, within clonal selection. Obviously, the B cell is going to take a lot of time to find its antigen when it's surrounded by 10 billion other B cells in a lymph node, surrounded by 10 billion other antigens. So it's like a mathematical possibility type of thing, and that takes time. In addition to the fact that that B cells are weird in a sense, and you also get to know that next year, in the sense that they need other cells to activate them, and that also takes time. And they, I'm pretty sure it's mentioned in your chapter, they also switch their types of antibodies. I'll explain that in a bit. That also takes time. There's so many changes that take a lot of time in the primary exposure that the body improves upon in the second exposure. And these include obviously naive lymphocyte activation. We already did that in the first exposure. We don't have to do that in the second exposure because the cell is not naive anymore. Does anyone remember what the definition of naive means?
Not sure, but I've never.
Exactly. So it's kind of like when you study all the theory, but you never have done practical. So you show up clueless. You don't know what you're doing. You're trying to find the right step. So these memory cells are not naive. They're competent. Obviously, they're immune competent, and they have immunological memory. So the instant they bind, they see the antigen in the bloodstream, they bind to it, they proliferate. So we have done this. We eliminated some time. B cell proliferation is much faster, and that's why I added the sonic here, because the B cell proliferation occurs much faster when it comes to the second exposure. And then you have the antibody production, which is also much faster, as your body has already learned or has been experienced to producing this specific variable region in the antibody. And we'll also see to that in a bit. And memory cell formation, we already formed memory cell. That's the one being activated in secondary exposure. So that's also these are the four things that kind of like are saved or kind of saving time within the second exposure. So that 3 to 6 lag no longer exists, right? So now let's look at what is the difference in the antibody response between primary and secondary exposures, specifically the antibody response.
What is needed about this?
Okay, that's one thing, but I want specifically when it comes to do with the antibodies themselves. Like, what changes do you observe from the first one to the second one?
More efficient.
Okay, more efficient, meaning what? What happens to the number of antibodies produced? Does it increase? Okay, right, correct. And what about their affinity to the antigen? What do you think happens? Exactly. So these are the two things you have to watch out for in the secondary, in the secondary, what's it called? In the secondary exposure. And you'll also know why that happens next year. So this whole chapter is a huge introduction to the big bomb that's going to be dropped on you next year when it comes to immunology in semester 1. So don't worry, no pressure at all, by the way. But it's going to be truly a journey. Right. So these are the 4 things. So you have greater affinity, like I mentioned, obviously. And you have the types of antibodies being produced is different. Okay, so I believe, I'm not sure if it's exactly mentioned, but it's important to know that in primary exposure, the first cells that are being produced, or the first antibodies, are IgMs. And IgMs are not that good because they're not specific. They're general. They're not specific. They don't bind with much affinity, and they're not the ones that usually have the same complement associated effects that I'll explain as well. that the other more specific or more specialized antibodies have, which are IgGs, IgAs, IgEs, whatever. I'll get to that also in a bit. So just keep this in mind that the types of antibodies in the second exposure is different. It's IgGs instead of IgMs. And even in the late primary exposure, it switches to IgGs. But after the first exposure, there is no more IgMs, all right? Increased antibody titers, like, what's your name? Noor, okay, like Noor mentioned, and more long-lasting titers. And this is explained by the graph here. So you see here in the first exposure to the antibody A, that antibody titer, the peak is lower, the effect lasting is way lower. You see it drops to basically nil very quickly, and the peak is lower and it drops to nil very quickly. But here, when it's exposed again, the peak is much higher and the slope is much higher as well. So the slope meaning the speed at which the antibodies are being produced, and you see that it doesn't immediately collapse to zero like it did here. It stays consistent and more prolonged. So this is stuff to watch out for in the secondary infection. Let's answer this question. T cells and B cells can be activated only by A, exposure to specific antigen. You can just say, we can just tell me the answer. Or B, pathogens. C, disease-causing agents. Or D, cells infected with viruses, bacteria, or cancer cells. Pardon? A? Yes, exactly. The answer is A. Why? Because A encompasses all the other options. It can be activated by antigens, by pathogens, because pathogens have antigens on their surface. It can be activated by cells that are infected with viruses or cancers, and also you'll know next year that the cells that are infected with viruses and cancers, they express different antigens, and therefore the body can recognize them, and the cancer and the viruses try to prevent that. It's a long story, and it's a very fun one as well. So yeah, just keep this in mind for now, that it's exposure to a specific antigen at a specific site in the plasma membrane is what triggers their activation. Now, select the correct statement about active and passive immunity. The antibodies utilized in active immunity are acquired from another organism and transferred to the host. Active and passive humoral immunity are both mechanisms of specific immunity that use antibodies. Vaccination is an example of introduction of passive, or immunological memory is established by passive immunization. Go on. B, active and passive humoral immunity are both mechanisms of specific, exactly correct. So this is a very important point. Just because it's passive, because you might think, oh, passive sounds weak, it sounds annoying, it sounds useless, that does not make it non-specific. You're still using antibodies, and antibodies are intrinsically specific, right? They're not innate, they're adaptive, right? And the only difference here is that active and passive is that passive is just, you don't get immunological memory, but you do have a specific response, right? So B is the correct answer. Great job. Okay, giving a patient an intravenous injection of immunoglobulin should cause them to produce antibodies to a pathogen. Trigger activation of memory B cells that make antibodies? No, that's wrong. D? Yes, temporarily protected from a specific disease. And why is that?
Because it's an artificial passive immunity, not active.
Exactly. So do you get immunological memory?
No.
Okay, so it won't be A because you won't produce antibodies, because producing antibodies implies that you have formed memory cells, because that only happens after clone selection and expansion, right? It doesn't protect them forever because there's no immunological memory, and it does not trigger activation, and it does not trigger activation of memory B cells because there's none being made. So, great job. Let's keep going. The type of resistance that is acquired as a result of developing and recovering from a disease is, yes, naturally acquired active immunity. This is very basic, and I'm using these questions to revise a specific aspect of the chapter because it's kind of like something you've already known from grade 10. We've all heard natural active, natural passive, artificial active, artificial passive. From grade 10, 11, 12, we're bored of it. So just any questions you find online, that'd be really helpful. What type of adaptive immunity will result from a mother to baby IgA transference? Naturally acquired passive immunity, exactly. Yes, so, and I'm going to use this, I'm going to talk about this later, alright? Next, naturally acquired active immunity is associated with which of the following? Is it exposure to a microbe? Naturally acquired active immunity? Is it transfer of IgCs from mother to fetus? Is it vaccination? Is it intravenous? Exactly, so it's A. Great job. So, everything, and you can just look at the options, you can find it from elimination. From the fetus, from mother to fetus, that's natural passive. Vaccination, that's active artificial. Intravenous is artificial passive. Alright, so what is the conclusion on the types of immunity, which are the ones that I've just mentioned? Active, meaning you get immunological memory. Active, natural, you naturally expose yourself to the disease. Active, actual artificial, vaccination, and you get immunologic memory. Natural, passive, from the same species or the same breastfeeding, and then you have artificial, passive, which is from another species or just intravenous IGs, right? Now, let's go to the basic antibody structure. This is really important because it's just really important to understand what's the distinction between all of them. So let's start with the antibody structure. So how many polypeptides are in an antibody? 4, exactly. You have 4 polypeptides in an antibody. You have how many light chains? How many heavy chains? Exactly. And the light chains and heavy chains are bound to each other by? Exactly. All right. And what is the region that binds to the antigen? What is it called? Okay, anyways, yes. So it's called variable region, and it's variable because it varies from every single B cell to the other, right? If they're not clones of each other, they're going to be absolutely different. And in your body, you have a huge, massive repertoire of B cells, and that's because of thousands of years of evolution and exposure to antigens and diseases, and obviously the heinous things that we humans do and expose ourselves to. So you're naturally bound to have an arsenal of B cells, right? And each chain has a variable region, but how many antigen binding sites do you have? You have 2. Exactly, you have 2. Because 2 on this side, 2 variable regions together form 1 antigen binding site, and on the other side it's the same thing. Now, what is the purpose of the constant region? Or where is it in the first place? Ignore the label. I said ignore the label. Where is the constant region? Yes, and it's the one that does not bind to anything, right? It does not bind to antigens in a sense. Obviously, it's going to bind to cell surface membranes in the case of B cells, mast cells, anything, right? So the constant region, and what is its purpose? Exactly, determines the class. So we have 5 classes, and they're determined by the FC region. Now let's explore these 5 classes. These are the 5 classes. I've conveniently used my emoji to kind of represent them. So the first one is 5 of them. So what is it? IgM. Now the second one is a dimer. Pardon? Exactly. The third one? G. Sorry? Yes, exactly. It's G. The fourth one. This is a lymphocyte. Lymphocyte and it's on the surface. D, exactly. And this is a rash. Alright, perfect. It is D. Now, why do we identify this with each other? Okay. Pentamer, defining character of what's it called? Of IgM, and this helps it in agglutination, as we'll see in a bit. You have IgA dimer, the only one present as a dimer, and then you have IgG, which is the one that transports it from the mother to the fetus. Keep in mind that there's a difference between mother and the fetus in utero compared to mother with milk. The milk antibodies are not IgGs, they are exactly. And here you have lymphocytes. Obviously, this one and this one are both present on lymphocyte surfaces, IgM and IgD. And this is not very clinically important, so I'm not going to explain it further. And here you have all to do with the allergic reactions, the parasitic infections, and all of that. And we'll see the mechanism as well later on. So, like I mentioned, you have this one, which is the pentamer, which is a potent agglutinating agent. And we'll see that in a bit. It's also very clinically significant when it comes to specific hypersensitivities and reactions and all of that, right? So this is one. And then I already mentioned that the monomer acts as a receptor on the B cells. Another important point is the fact that it activates complements. So what does it mean? So we know that there are 3 different pathways of complement activation, right? Do you guys know that? Pardon? They canceled it? Okay. You're going to know them next year. So just from a small heads up from now, is you have the classic pathway, which is where it's activated by IgGs and IgMs. So they are the ones that activate the complement. So just keep that in mind. The way I remember it is GM, General Motors, activate complements. Like General Motors, you know the car brand General Motors, or I don't know, whatever they do it nowadays. They're a very irrelevant company. So yeah, and then I already mentioned that they're the first gamma globulins that are being produced in the primary response, and they're never produced again after that when it comes to that specific antigen. Alright? Okay, now we move to IgAs, which are for mucosal immunity. So, skin, gastric mucosa, respiratory mucosa, milk, any bodily secretions that is exogenous has IgAs. Remember, it's that. And I highlighted milk here because just because it's helping the baby after birth doesn't mean it's helping it before birth. Before birth, the antibody that's traveling across the placenta is? Sorry? Exactly, correct. And another thing about IgA is that it prevents from reaching epithelial surface, like I mentioned. Now, moving on to IgGs, which are the most important antibodies and they're the most clinically significant, right? They are the ones that are being produced in every infection after the initial burst of IgM in the early primary phase. After that, we do not get IgMs, we get IgGs. Complement activation, like I just mentioned as well. In addition to it crosses the placenta and gives the baby immunity, and that's why you notice that you won't immediately, if a baby has a congenital autoimmune disease or a congenital immune deficiency, they won't present with it to the hospital until a few months afterwards, because the IgGs and the IgAs in the milk protect them from all these diseases. And it's the most abundant antibody, and that's very important. And last but not least, we have IgEs, and they're the most significant in mediating allergic reactions, like I mentioned. And the whole mechanism is actually quite interesting. So I'll explain it right now, so that when I get to hypersensitivity type 1, I don't have to explain it. So what happens essentially is that your body is not intrinsically allergic to anything. Your body has to be sensitized to an allergy. So, for example, let's say I'm allergic to peanuts, right? And I consume peanuts for the first time in my body, and my body decides to have a problem with it. So it recognizes it, but it does not initiate a response against it. And this results in the release of IgEs from mast cells and eosinophils, and all these cells are responsible for allergic reactions, right? So these implant themselves on the constant region implants itself into where? Into the cell surface membrane of the mast cell and the eosinophil, and the antigen binding, or the binding site, it protrudes outwards. So that the next time I eat peanuts, these will bind, and immediately the eosinophils and the mast cells will degranulate. Degranulate meaning release all their histamine, and that gives you an allergic reaction. Do you guys get that? So that's basically how type 1 hypersensitivity happens, and we'll get to that in a bit. So now, antibodies secreted from the mother's milk are what? Yes, correct. Alright, and which of these antibody classes is often arranged as a dimer? Yes, exactly. So IgG, is it a dimer, pentamer, solomer? Monomer, exactly. IgM? And when is it a monomer? Exactly, when it acts as a receptor in B cells. And it's not all of the above, obviously. So now, I have a matching question for you guys. So match the following immunoglobulin classes to the appropriate characteristics of function. So, current infection. So current meaning your first ever exactly. B cell receptor. D, okay, and? Yes, exactly, correct. Crosses placenta? Activates complement? And? Correct. General motors, keep that in mind. And mucosal immunity? Correct. Next. So now we know antibodies, we know what they do, each class. Now we have to know how they act, how they do their function, right? Okay, so it's a good mnemonic mentioned in the book. I didn't create it. It's called PLAN. So PLAN is essentially 4 steps, or 4 different ways that antibodies can do their effects. P, which is precipitation, which means it binds to soluble antigens, so many of them, that they cross-link and make such a big compound that is insoluble, and that makes it easier for the phagocytes to digest them. It's important to note that every single one of these promotes phagocytosis and promotes inflammation, because B cells don't have a cytotoxic function. B cells are purely messages, they're purely markers, they're purely intermediates. They don't do anything except warn and bring other cells that can actually destroy the antigens, the cells, the viruses, the bacteria. So this is important, and just to understand, because next year you're going to be dealing with a lot of these, so don't be confused with it. So you have precipitation, you have lysis, which is mediated by, exactly. And complements do multiple things. So specifically, there's a complement called C3b, you don't have to know it. It opsonizes. It opsonizes, meaning it tags and the B cells, the pathogens of the viruses, and makes them easier to target by the phagocytes. And it does lysis, meaning punching holes. They group together, they form something called MAC. What is a MAC? Is it not in your book? I think think I'm more preaching year one than I am preaching foundation. All right. So basically, it forms something called membrane attack complex, and that punches a hole in the cell membrane, and that leads to lysis of the cell. And then you have, last but not least, you have agglutination, which is mostly significant in IgMs because they have 10 antigen binding sites, and so they can bind to a lot of different antigens and get everything together, and that forms big antigen-antibody complexes, which leads to more phagocytosis, more inflammation, and last, neutralization. It binds to potential antigens that could get the bacteria into the cell. So to prevent that from happening, we have antibodies. So these are the 4 actions. And now for some questions. The process of coating a microbe with antibodies to make it more sensible. Exactly, correct. Which of the following antibody capabilities causes transfusion reactions? Correct, good job. This is also very significant in terms of pentamers, because pentamers, the pentamer IgMs, are the ones that mostly communicate that. And which of the following mechanisms of antibody? Good job. I don't even have to say the question. All right. Now let's go to the clinical implications. So this is where we kind of have to worry about the Superman here, because he's kind of just in a tough spot in clinical implications. In some, he's the villain, in some, he's the victim, and we're going to find out which is which in a bit. So the 4 clinical implications that we have to cover as part of your curriculum is monoclonal antibodies, which were mentioned by my colleague Noor, I believe, and it's basically a type of antibodies that are used for immunotherapy. So what is an immunotherapy? It's a therapy that uses the immune system, specifically the antibodies, in order to treat infections. It can be used to treat cancer. You don't have to know this, just know that it's used in diseases, or specifically when you lack specific antibodies, and these are complementary to the antigen that's invading your body. And that's actually what happens when you get an intravenous IV. You're injecting into intravenous, IgGs. You're injecting monoclonal antibodies, right? And then we have graft rejection. This has a whole lecture on its own next year, and you'll see why. But just for now, know that there's different types of grafts. You can graft yourself before your surgery. You can take out a part of your own tissue and then use it to graft to yourself later after that. That's called exactly. And then if it's if someone else donated their skin or their organ to you, what is that called? Yes, xenograft. And if it's from a pig or from another animal, xenograft, right? And the possibility for rejection increases dramatically when we stop using autografts. Isografts, there's possibility of rejection. That's why we match with families before we do organ transplants, and xenografts are like kind of dumb, because why would you want a pig in your body? Like, no one wants that. So just keep that in mind. And lastly, before we get to the biggest topic, which is the autoimmunity and hypersensitivities, we have to discuss immune deficiencies. Immune deficiencies can be acquired or they can be congenital. So what's interesting is that both of the diseases mentioned here have their own weeks dedicated to them next semester. So you have a whole week next semester about HIV and a whole week about SCID. And you'll see why when you do the respective immunology for them, right? So in SCID, the patient or the infant is born with very, very little T cells. And I don't know if you've read past your included parts, but you'll know that T cells activate B cells. So if you have a problem with T cells, you're going to have a problem with B cells. And so everything's going to go to trash after that. So everything's going to go to trash after that, and that's why you have SCID. For the case of HIV, HIV infects T cells as well. And like I said, T cells activate B cells. So if you have a problem with T cells, it's going to have another problem. And here in this case, HIV is a virus, so it's acquired. And we know how it's acquired, mostly by bodily fluids, unprotected intercourse, all of that. Right? So now moving on to the most important clinical implication, which is autoimmunity and hypersensitivities. So what is autoimmunity and why does it happen? So can everyone tell me?
Oh, your body makes autoantibodies.
Right.
So failure of self-tolerance.
Yes. Why do you have a failure of self-tolerance?
The regulatory mechanisms that normally keep in check your self-reactive lymphocytes.
Okay.
So this can happen.
All right, but there are specific mechanisms that you can become autoimmune, right? So do you have in mind any of these mechanisms? No? Okay, it's all right. This again has a whole lecture dedicated to it next semester, and you just have to know that there are some mechanisms where there is some foreign antigens that resemble your own antigens. And therefore that causes a collapse of your body recognizing your own antigens, and it starts to invade them, confusing that could be a foreign antigen because it looks like the allo antigen or the foreign antigen, right? And another scenario, which is where your new self-antigens appear. And the importance of this is that your cells, your B cells while they're maturing and T cells while they're maturing, they're exposed to a lot of antigens in your body. But there is some that are not exposed to it. So in the case where there is trauma in these sites where these antigens are, that exposes the antigens that were not exposed to the T cells and the B cells during maturation, the body will not recognize them and be like, oh, this is a foreign antigen, and so you have autoimmunity. And one of these very interesting things is the eye. Like, I'm pretty sure you guys must have stumbled into an Instagram reel that says, did you know that if your body knew your eyes existed, it would kill them or you'd go blind? And that's because a lot of antigens in your eye were never exposed to your T cells while they were developing, and your body starts to kill them instead, right? So this is important, and how do you treat them? Well, you all know what these mean because these are more a reference to me than it is to you to know the answer. But how do you treat autoimmunities? Yes, you can do that. You can do that. You can do that, but that's a very general answer. Immunosuppressants target specific mechanisms, right? So they target and block co-stimulator molecules that activate B cells, and they prevent the release of inflammatory cytokines that promote more inflammation and more autoimmune reaction. And when you finish year 1, semester 1, you'll understand what this means when I wrote it. So this is kind of like a Morse code for you guys now, but you'll get to know it eventually. Right? So now we start with the final part of the chapter, which is hypersensitivities. The easiest part. IG type 1, I explained it already, involves IG. Which IG? IgEs. First exposure, no response. Second exposure, IgEs are already present on the surface of mast cells and eosinophils, I think. Basophils, yes, basophils mostly. Because they're the ones with histamine, and then histamine is being released, you get an inflammatory reaction, and it's immediate. So immediately after you eat peanut butter or egg whites or dates, you would have this reaction. And it's important to note that type 1, 2, and 3 all involve antibodies. Type 1 involves IgE, type 2 and 3 involve IG, M and G, mostly G, and type 4 involves cytotoxic or just T cells generally, right? So this is for type 1. Moving on to type 2, which is cytotoxic. So when I was in foundation, no one really explained to us the difference between type 2 and type 3 very clearly. So I'm here with my year 1 knowledge explaining to you what the difference is. So type 2 involves the antibodies binding to antigens on cells. Type 3 involves antibodies binding to free antigens that have no cells attached to them. And the most famous example of a type 2 hypersensitivity, well, it's not really famous at all. There's 10 billion other examples that are way more clinically relevant. But this one is the one that's mentioned in your curriculum, which is a transfusion reaction, which is where blood transfusions go, you have a bad typing, you have a bad, you don't have a match of blood groups. I'm pretty sure you guys took it in the chapter. So the antibodies with the superhero lymphocytes bind to the surface of the cell, and they form complexes. And these complexes, they don't really form complexes. They stimulate complement-mediated lysis. We already said IgGs activate. Exactly. What's the other one? Activate complements? Exactly. Correct. So this is how it works. And obviously, you're gonna have complement-mediated lysis. You're gonna have the lysis and the opsonization that we spoke about. Next, type 3 immune complex, free antigens binding to not really free antibodies, but free antigens binding to antibodies on the surface of cells. And these form immune complexes. Immune complex is basically just a mess of antigens and antibodies that are bound to their lymphocytes. And this is really important because when these immune complexes deposit in your body, they cause inflammation. And that's why when you have, has anyone ever heard of the disease lupus? Yes, so lupus is a very famous example of a type 3 hypersensitivity. And if you knew anyone with lupus, you know it causes a big red rash. And this red rash is a product of the damage to the blood vessels that occur under the skin because of this hypersensitivity, because of the immune complexes depositing in the walls of the blood vessels causing inflammation. So this is really important. And usually in type 3 hypersensitivities, you would see some sort of systemic inflammation going on. But that's beside the point. Just stick to the fact that it's an immune complex deposition which causes inflammation when it deposits. Last but not least, we have type 4 involving which cells? Exactly, T cells. They're both T cells, and it's delayed. So type 1 immediate, seconds or minutes after you eat it. Type 2 and type 3, hours, maybe a day. And then for type 4, it's completely delayed, multiple days, right? Multiple days. And the most famous example is, yes, which causes specifically, yes, atopic dermatitis. That's a very important example because the poison ivy enters your body and it binds to what? It's an antigen, but it binds to your self-antigens, right? And when it binds to self-antigens, it's called a hapten and produces a new antigen, which is recognized by your body as foreign. And this is why you get type 4 hypersensitivity. And this is very common in, for example, people who wear jewelry quite often, people who go to the forest, explore with poison ivy. And this is very clinically significant as well for tests for tuberculosis and stuff like that, right? So this is type 4, and we are done. We have a few questions, and then we'll be completely done. So, a 25-year-old who was recently diagnosed as a patient of AIDS, or acquired immune deficiency syndrome, which is the following: it happens due to HIV infection. Correct. When I first saw the question, I was like, "What the hell is this?" But then I realized that AIDS is AIDS, and AIDS is caused by HIV. So it's kind of a mess. So be careful. A 40-year-old woman presents to the allergist with a history of recurrent episodes of abdominal pain, bloating, and diarrhea. She reports that the symptoms occur shortly after consuming dairy products. Shortly after consuming dairy products. It is type 1 hypersensitivity. Great job. What's the dead giveaway? Exactly, correct. And also because it's food. Food is mostly the cause of type 1. 25-year-old woman presents...
D.
Did you do this previously?
No.
Are you sure?
Yes.
Alright, how did you know that it was D?
Itchy patches a few days.
Red itchy patches. Itchy patches, skincare products? Right. So she has a few days afterwards, it would be yes, it is type 4. A few days. So our last question and last slides for this session, hopefully you guys enjoyed my part, was a matching question. And it's important to note that matching, as you guys probably have witnessed in the midterm, I believe, that they love matching questions in Bio 2. And they're usually pretty easy. They're very easy marks to score, which is why I personally love them, because you can have a lot of elimination going on. So, will you tell me what this answer is? Yeah, it's C. Yes, I remember it being C. Yes, exactly. So, allergies immediately is 1. Okay, so it's not B or D. SLE, systemic lupus erythematosus, which is 3. Anaphylactic shock. Exactly. Do you guys know how, you guys did cardio, I assume, which mentioned anaphylactic shock. So you know that is type 1, it's immediate. And type 4 is allergic reaction after a few days, which is type 4. And that's it for this session. I hope you guys enjoyed and thank you so much.